Welcome to the deep dive. Today we're plunging into a world that's, well, fundamental to every living thing on this planet, yet often goes unnoticed. The constant dynamic dance of water and salt within animal bodies. To kick us off, imagine a blue crab. It's a really dramatic, visually striking example of something crucial happening just beneath the surface of, well, all animal life.
Exactly. We're talking about maintaining a stable internal environment, the concept called homeostasis at its core. But as we'll discover, animals have evolved these incredibly diverse and honestly often surprising strategies to achieve it.
Okay, let's unpack this. Our mission today is basically to cut through the complexity. We want to give you a shortcut to understanding how animals, from tiny desert mice to mighty ocean sharks, manage their internal water and salt balance. How they survive and thrive in wildly different environments. We'll be digging into key insights, concepts, mechanisms, and some great real-world examples from a top textbook, Animal Physiology by Hill, Wise, and Anderson. And that blue crab is just a perfect starting point, isn't it, to show how dynamic this all is. So picture this. A blue crab, Calanectes sapidus for the biologists out there, needs to shed its exoskeleton to grow. It does this more than 25 times in its short, 2-year life, which is amazing in itself. When it's ready to molt, it takes on this massive amount of water from its environment, sometimes increasing its body weight by 50%, maybe even 100%.
Yeah, and that isn't just about getting bigger, you see. It's a really strategic move. This rapid water intake serves two vital purposes. First, the swelling physically cracks open the old, rigid exoskeleton. It lets the crab literally pull itself out. Second, by immediately inflating to a larger size, it creates the space needed to start synthesizing a new, bigger exoskeleton right away.
Ah, I see.
Later, as that new armor hardens, it'll void the excess water, sort of growing into its new shell.
So the blue crab essentially weaponizes water for its growth. Okay, what does this dramatic example teach us about water exchange in all animals, then?
Well, it highlights a crucial, often overlooked principle. Water exchanges are incredibly dynamic in every animal. Think about a mouse. It turns over about 25% of its body water daily.
25%? Wow.
Yeah. Humans, being larger, turn over maybe 7%. But even that 7% means your body weight could fluctuate by several kilograms, maybe 5 kilograms, in a single day, just from water imbalance.
Yet, despite this constant flux, healthy animals maintain a surprisingly steady body water content, most of the time anyway.
Precisely. They possess these highly responsive mechanisms that perfectly match water gains and losses. The crab's molting is a controlled, temporary deviation from this fundamental balance. It deliberately unbalances things for a specific purpose, before resuming its tight regulation.
Okay, so given this rapid turnover, what is this critical internal fluid environment we're talking about?
Well, the sheer abundance of body fluids tells you a lot. They account for more than half an animal's body weight in most cases, around 60% in adult humans, I think, and maybe a staggering 95% in certain jellyfish and things like that, gelatinous invertebrates. These fluids are quite literally the immediate environment for all cells, their internal structures, proteins, molecules, everything. Their composition is the context for all body functions.
And what are the key players in these fluids? What makes them up?
Primarily water and inorganic ions, which, most of us know as salts or electrolytes. These inorganic ions like sodium, chloride, potassium, they're body's hidden architects and electricians, you could say. They literally sculpt proteins into the right three-dimensional shapes. And they're the spark plugs for nerve signals, muscle contractions. That's why things like too little intracellular potassium can severely alter heart rhythm. It's critical stuff.
And water itself isn't just a passive solvent here, is it? It plays a more active role than we often assume.
Absolutely. Water is the essential matrix, yes. But its amount directly influences ion concentrations. It affects cell and tissue volumes. And its movements, driven by osmosis, create hydrostatic pressures within the body. What's really fascinating here is our growing understanding that water itself has a complex structured presence around ions and macromolecules. We call them shells of hydration. These structured solutions, with their intricate hydrogen bonding, well, they have physiological implications we're only just beginning to fully explore.
Wow, so water isn't just simple H2O in the body. It's almost like it has its own complex architecture. And these fluids exist in different compartments you mentioned. How do they interact? Is it a constant flow or more selective?
Oh, there's a brisk, constant exchange. We divide them into three main compartments. Intracellular fluids, inside the cells. Interstitial fluids, that's between cells and tissues. And blood plasma, the liquid part of blood. They're incredibly close together, separated only by cell membranes or the thin layer of capillary walls. Water and ions move readily between them. Osmosis, diffusion, active transport, even bulk flow driven by blood pressure. It's very dynamic.
Sounds incredibly complex to study directly. Physiologists must have a go-to method, right, for getting a snapshot without, poking every cell.
They do, and it's surprisingly practical. They almost always sample only the blood plasma. It's relatively easy to collect. And what's insightful is that blood plasma gives us a great deal of information. Why? Because the interstitial fluid is typically very similar osmotically, due to that free exchange we talked about. And intracellular fluid usually matches the osmotic pressure of the interstitial fluid and plasma too. So plasma osmotic pressure tells you a lot.
Okay, so osmotic pressure tends to be similar across the board.
Generally, yes. But the crucial distinction, though, is that the ionic composition inside cells differs dramatically from plasma. That's largely thanks to the tireless work of ion pumps within the cell membranes.
Ah, right, the pumps. That's a key distinction. So we've seen how dynamic these fluid exchanges are, how critical the internal environment is. But how do animals control it all? What are the fundamental strategies they use to keep this delicate balance?
At their core, animals either regulate or they conform. Regulation means actively maintaining a constant or nearly constant internal condition. When we talk about blood plasma, there are three main types of regulation. Osmotic regulation, keeping osmotic pressure steady like humans around 300 millismolar. Then there's ionic regulation for specific ion concentrations and volume regulation for the total amount of water, like maintaining blood volume. Humans, yeah, we do all three.
And if regulation takes constant effort, constant energy, what's the alternative?
That's conformity. In this strategy, the animal just allows its internal conditions to match the external environment. A blue mussel, for instance, is an osmotic conformer. Its blood osmotic pressure simply tracks whatever the water around it is doing.
But it's not always so black and white, is it? Animals can kind of mix and match.
They certainly do. You see a whole continuum in nature. A marine shrimp might be an almost perfect osmoregulator. A green crab, on the other hand, regulates its blood osmotic pressure really well in dilute waters. But if the water gets more concentrated than seawater, it then becomes an osmotic conformer. Switches strategy.
Interesting. It seems like these three types of regulation, osmotic, ionic, volume, they must be really tangled up together often. Like you said, a freshwater fish taking on water by osmosis, that's going to challenge all three at once, right?
That's a great observation. And yeah, it can create the illusion that they're all the same thing. But they're distinct processes, and understanding that distinction is a crucial analytical tool for physiologists trying to figure out how animals manage things. The adaptive significance, the why, behind choosing regulation versus conformity really highlights this trade-off.
So what does this mean for survival? What are the pros and cons?
Well, regulation costs energy. Animals have to invest metabolic energy to maintain that constancy. But the payoff is huge. It gives their cells a stable osmotic ionic environment, which is a big advantage for consistent cell function. Conformity, on the other hand, saves energy because the animal isn't actively fighting the environment. But its cells then have to be able to cope with whatever changing conditions the environment throws at them. It's a fundamental biological trade-off, really.
Let's talk about the environments themselves then, because those challenges are what drive these strategies. What about aquatic environments?
Well, the open ocean is remarkably uniform and stable. Salinity around 34-36 grams per kilogram. Osmotic pressure almost exactly 1,000 millismolar. Very predictable. Freshwater, though, is the opposite. Very low osmotic pressure, usually between, say, 0.5 and 15 millismolar. But even there, specific ion concentrations, especially calcium, can vary a lot and really impact physiology.
And then you have the dynamic in between places like estuaries.
Exactly. Brackish waters, where ocean meets fresh water. They're physiologically fascinating places. Estuaries, like Chesapeake Bay, are prime examples. Salinity can swing dramatically, sometimes from less than 5 grams per kilo up to over 25 over relatively short distances. And blue crabs, again, are highly effective osmoregulators in these variable brackish conditions, well, when they're not molting. Their blood osmotic pressure stays almost constant, even as the outside salinity goes up and down like crazy.
Okay, and what about life on land? That must present a completely different set of challenges, especially around water loss.
Totally different. Terrestrial animals are surrounded by air, which only has water as a gas and is basically salt-free. The atmosphere is almost always trying to pull water out of the animal. Animals primarily lose water to it by evaporation. Living in air is just inherently dehydrating, which makes deserts obviously really important places for studying water stress. They're the extremes of terrestrial water challenges, even if they aren't necessarily hot all the time.
So evaporation is the big enemy. How does it work for an animal and what controls the rate?
Evaporation is basically a special case of gas diffusion. Water always diffuses from where its vapor pressure is high to where it's low. So if an animal's body fluid is in contact with the air, net evaporation happens if the water vapor pressure in the body fluid is higher than in the air, which it usually is. The rate increases with that difference. So warmer body fluids, drier air, more wind, all increase water loss. It's a constant battle.
So the animal's main defense is basically how waterproof its skin is.
Precisely. What physiologists call low integument permeability, essentially. How good their outer covering is at stopping water from getting out. That's the chief physiological defense against drying out on land. As for gaining water from the air by condensation, it's pretty rare. Generally only happens if an animal's body surface is cooler than the air, like, an iced tea glass on a humid day. Maybe lizards coming out of cool burrows might gain a tiny bit temporarily, but they warm up fast and then start losing water again.
Right. So how do animals actually do this regulation? Which organs are the main players in these balancing acts?
Well, the kidneys play hugely important roles across most animal types. Their fundamental job is to regulate the composition of the blood plasma. They do this by removing water, salts, and other solutes in controlled amounts. Gills are also crucial in aquatic animals, obviously, and salt glands are key for certain birds and reptiles that need to get rid of excess salt.
And physiologists use a handy conceptual tool to understand kidney function, UP ratios. Can you explain those? Why are they so powerful?
UP ratios. They're simple but incredibly powerful, yeah. It's just the ratio of a substance's concentration in the urine to its concentration in the plasma, P. For osmotic regulation, we look at the osmotic UP ratio. If UP equals 1, the urine is isosmotic plasma. The kidneys can't directly change the plasma's osmotic pressure in this case. If UP is less than 1, the urine is hyposmotic, more dilute than plasma. This preferentially gets rid of water, which helps raise the plasma osmotic pressure.
Like freshwater fish.
Exactly, counteracting water influx, or us, after drinking a lot of water. Conversely, if UP is greater than 1, the urine is hyperosmotic, more concentrated than plasma. This preferentially dumps solutes, which helps lower plasma osmotic pressure. That's what mammals, birds, and insects do to manage high internal concentrations or conserve water.
This really highlights that point you made earlier, that osmotic, ionic, and volume regulation are distinct jobs. How do kidneys show that independence?
They illustrate it perfectly. Take freshwater crabs again. Their kidneys are vital for volume regulation, constantly pumping out the excess water they gain by osmosis. But their urine is always isosmotic to their plasma. UP is always 1 for osmolality. This means their kidneys play absolutely no direct role in osmotic regulation itself. They regulate volume, yes, but not osmotic pressure directly.
Fascinating. So they can specialize. And ionic regulation can be separate too.
Yes. Marine teleost fish, bony fish in the sea are a great example. They live in salty water, saltier than their body fluids, so they constantly lose water and gain ions. Their kidneys produce isosmotic urine, UP equals 1 again. So no direct help with osmotic regulation there either. But their urine has very high UP ratios for specific ions like magnesium, sulfate, calcium. By actively pumping these excess ions into the urine, ions they gain from seawater, their kidneys play a crucial ionic regulatory role, completely separate from osmotic regulation.
Okay, so beyond kidneys and direct exchange with the environment, where else do animals get water and salt or lose it? Diet must be a huge factor.
Oh, absolutely huge. The specific composition of food and drinking water has major implications. Think about marine predators. A marine mammal or fish eating invertebrates, which are isosmotic to seawater, remember it takes on a massive salt load, they have to work hard metabolically to eliminate that excess salt. But if they eat other fish, which are hyposmotic to seawater, much like themselves.
Less work for them.
Right. They incur very little salt load. It's a cool lesson in ecological energetics. The fish-eating predator basically benefits from the work its prey already did to keep its own body fluids dilute.
And the famous paradox, water everywhere, nor any drop to drink. Why does drinking seawater dehydrate humans?
It comes down to our kidneys' limits. Our kidneys simply cannot produce urine that's concentrated enough in chloride ions. Seawater has a higher chloride concentration than the maximum our kidneys can possibly achieve in urine. So if we drink seawater, we'd actually use more of our body water to excrete the chloride we just ingested than the water we actually took in. Net loss, dehydration. Some animals, though, marine birds, reptiles, some mammals, can excrete salts at much higher concentrations and can gain net water from salty solutions.
And what about plants? Can eating plants be a salty problem?
Oh, definitely, especially in deserts. Imagine a desert plant that accumulates so much salt. Its tissue fluids are even saltier than seawater. These are called halophytes. For most animals, eating them would be like drinking seawater instant dehydration. But astonishingly, creatures like the desert sand rat can thrive on them. Their kidneys have these, well, legendary concentrating abilities. They can produce urine so salty they actually extract net water from these hyper-salty meals. It's just mind-blowing adaptation.
Wow. And even dry food isn't always truly dry, is it?
That's a good point. Air-dried foods, like barley grain, still contain moisture, and the amount varies with the surrounding air humidity. This is really significant for desert animals. They can actually increase their water intake by feeding at night when humidity is higher, or by storing food in their burrows, where the humidity is also generally higher than outside. And, maybe surprisingly, protein-rich foods can actually be dehydrating for terrestrial animals. Why? Because when animals break down protein, they produce nitrogenous wastes, like urea in mammals, that require water for excretion. So a high-protein meal can force an animal to use up more precious water to get rid of that extra urea compared to a low-protein meal.
So food isn't just nutrients, it's a massive part of the whole water-salt equation. But there's one more source of water, isn't there? The hidden one, metabolic water.
Ah, yes, metabolic water. This is crucial. It's the water produced chemically directly from the aerobic breakdown, the oxidation of organic food molecules like glucose. It's produced by all animals that breathe oxygen. But, and this is really important, you have to distinguish between gross metabolic water, the total amount produced, and net metabolic water. To get the net gain, you have to subtract the obligatory water losses that happen because of that metabolism. Things like respiratory water loss through breathing, urinary water loss to excrete the waste products, and fecal water loss.
And the classic example here is the kangaroo rat, right? The one that doesn't drink.
Exactly. Kangaroo rats can thrive in deserts, just eating air-dried seeds like barley, without ever drinking liquid water. Now, the common myth is that they produce more metabolic water than other animals, but that's not actually true. All animals produce roughly the same amount per gram of food oxidized. Their secret isn't making more water, it's exceptional water conservation. They have incredibly low urinary losses because their kidneys are amazing at concentrating urea, and they have very low fecal losses because they produce extremely dry feces. So their net gain of metabolic water is small, maybe just plus 0.07 grams of water per gram of barley eaten, but because their losses are so tiny, that small net gain is critical for survival.
So metabolic water matters most in animals that are already really, really good at conserving water overall.
Precisely. For an animal that conserves water incredibly effectively, like the kangaroo rat, its metabolic water production becomes a large and vital part of its total water budget. But for an animal that loses water easily, that same amount of metabolic water production would be insignificant compared to its huge daily water turnover. Context is everything.
Okay, let's zoom right in now, down to the cellular level. Cells need to maintain their volume, but their membranes let water pass through easily. How do they cope if the fluid around them changes concentration? Seems like they'd be constantly swelling or shrinking.
It is a constant challenge, absolutely. If the extracellular fluid's osmotic pressure changes, water will move across the cell membrane, causing cells to swell in dilute environments or shrink in concentrated ones. Neither is good for function. So, to regulate their volume, cells have to change their internal content of osmotically effective solutes, dissolved stuff. If the outside fluid gets too dilute, they need to reduce their internal solutes. If it gets too concentrated, they need to increase them. And this also highlights, again, the adaptive significance of osmoregulation at the whole animal level. If the kidneys or gills or whatever keep the extracellular fluid osmotic pressure constant, then the cells are basically freed from having to constantly fiddle with their own internal solute levels.
And this is where organic solids come into the picture in a big way, right? It's not just about pumping in organic ions like sodium and potassium inside the cell for this.
Exactly. This is where it gets really interesting, and it's been a key evolutionary insight over the years. Animals are quite selective about which solutes they use for this intracellular volume regulation. Many invertebrates, like the mussels and blue crabs we mentioned, and even specific vertebrate tissues like the brain and parts of the kidney, primarily use organic solids. Things like free amino acids. They can adjust the levels of these amino acids by changing how fast they break them down, or make them, or break down proteins, or even by actively transporting them. You can actually see this happen. If you move some marine invertebrates to more dilute water, you often see a spike in ammonia excretion.
Ah, breaking down amino acids.
Exactly, they're catabolizing amino acids to reduce the internal solute concentration and present swelling. And interestingly, The big difference in total intracellular solute concentration between, say, a typical vertebrate and an open ocean invertebrate isn't mainly due to inorganic ions. It's mostly down to these varying levels of organic solutes. It allows for simultaneous cell volume and ionic regulation.
So clever. Okay, this brings us to the molecular players. Osmolytes, compatible solutes, and counteracting solutes. Can you break those down? Why are these distinctions important?
Sure, an osmolyte, or sometimes called an osmotic effector, is basically any solute that affects osmotic pressure and that the animal actively adjusts for regulation. Could be inorganic or organic. Now, the reason animals often prefer organic solutes as intracellular osmolytes leads us to the concept of compatible solutes. These are special solutes that, even when they build up to high concentrations inside the cell, have minimal disruptive effects on important proteins and macromolecules, like enzymes. They can be increased or decreased quite a lot without messing up how enzymes function. Examples include certain free amino acids like glycine, proline, taurine, and things called betaines, even glycerol in some cases. This really explains why animals lean on these organic molecules for cell volume regulation. They're safe for the cell's machinery.
Okay, so compatible solutes are safe to have around in high amounts, but then there's another layer. Counteracting solutes. What's that about?
Yes, another layer of biochemical elegance. Counteracting solutes are solutes that, if present individually, might actually disrupt macromolecules. But they occur in teams of two or more that work together to offset each other's negative effects. As a team, they become relatively harmless. The classic textbook example comes from sharks, skates, and rays the elasmobranch. They use urea as a major osmolyte to match seawater concentration. But high urea on its own tends to destabilize proteins, mess with their structure and function. However, these fish also accumulate methylamines, particularly trimethyl monoxide, or TMAO, and TMAO does the opposite. It tends to stabilize proteins. So the stabilizing effect of TMAO effectively counteracts or titrates away, as chemists might say, the destabilizing effects of urea. This allows sharks to maintain incredibly high internal urea concentrations, essential for their water balance, without poisoning their own enzymes.
Wow, so it's not just about what chemicals are in the body, but how they interact almost like a biochemical tag team to keep everything running smoothly. That's a truly elegant solution to a fundamental biological problem. So, let's try and wrap this up. What does this all mean? We've taken a pretty deep dive into this incredible, often hidden world of animal water and salt physiology, from the, really dramatic molting of a blue crab to the microscopic dance of molecules inside our own cells. We've seen how animals are constantly adjusting. They're maintaining this delicate balance of their internal environment, whether they're stuck in the deep ocean, a freshwater river, or the absolute driest desert.
It's truly amazing how life adapts, isn't it? And like you said, it's not just about drinking or peeing, the obvious things. It's about this intricate interplay of fluid compartments, these surprising strategies of regulation versus conformity, and these remarkable molecular tricks like compatible and counteracting solutes. All of this allows life to really flourish in pretty much every corner of our planet. Understanding this physiology gives us such profound insights into adaptation, evolution, and survival.
Absolutely. And it leaves us with maybe a final provocative thought for you, the listener. Consider the sheer interconnectedness of it all. The water a kangaroo rat gets from its seemingly dry barley seeds, the specific salts a shark uses to make urea tolerable. These aren't just isolated biological facts. They're part of a grand, evolved strategy, a testament to how life rises to meet every conceivable environmental challenge. What other surprising physiological strategies might be hiding out there in plain sight, just waiting for their own dip dive? A big thank you for joining us today on the Deep Dive and being part of the Last Minute Lecture family. We hope this journey through animal water and salt balance was as fascinating for you as it was for us.